So today we are going to discuss new chapter from your biochemistry. Up to today we have already seen biochemistry of your carbohydrates, biochemistry of lipids, metabolism of carbohydrates, metabolism of lipids. Then we have seen the proteins and amino acids. We have also seen the metabolism of proteins and amino acids. So again I would like to take one more session on proteins and amino acids. But whatever is important for your GPAT and all other exams, we have already done with that. So whatever additional points I am finding from various books, now that points will be presented in the next lecture in front of you. So that is nothing but the metabolism of individual amino acids, then some amino acids and their IPAC names, then amino acids, okay, then and their IPAC names, then diseases or diseases related to protein deficiency, right? Like marasmus, kwashiorkor. These two are the protein deficiency disorders. So that will be discussed in the half an hour or 45 minutes in the upcoming sessions. But today we'll see nucleic acid. Again, very important topic from your biochemistry, because biochemistry consists of biochemistry is a very important subject, which is a basic subject, and if you have knowledge of biochemistry, definitely you can understand your life. So today we'll see biochemistry of nucleic acids. As the name indicates, nucleic acid. So nucleic acid means what? They are found in the nucleus. Nucleic acid means they are found in the nucleus, and they are very important for the hereditary character of humans. With nucleic acid, so they are obviously found in the nucleus. You know that there are two nucleic acids, DNA and RNA. They are the okay. So every human being is a different, has a different genetic makeup. Seldomly you will find two humans which are looking exactly like the same, but everybody has different genetics. Everybody has different genes. That's why everybody has different physics and as well as the biochemical activities in the body. So everybody has different physics, biochemical activities. Everybody is having different looks, and because of everybody has different genes. Everybody has different genes. Everybody has different DNA. Now, nucleic acid. There are two type of nucleic acid. You know that DNA and RNA. DNA means deoxyribonucleic acid, and RNA means ribose nucleic acid. So, whatever important points from this individually. First of all, there are nucleic acid, or they are also known as genes. Now, the genetic information which are present in these genes in the DNA, whatever genetic information is there, which is passed from your parents, which is passed from your grandparents to you, or to your parents, from your parents to you, whatever genetic material is there, whatever genetic information is there, whatever genetic makeup is there, so that is inherited. That is inherited from your grandparents to your parents, from parents to you, and from you to your children. So that is why the information is resided, the information is stored, the information is found, information is present on the genes. This is DNA. So DNA or nucleoproteins, they are also known as nucleoproteins, nucleic acids. Okay. Now these nucleic acids, they are present in the nucleoprotein. Nucleic acid is a component of nucleoprotein. Now nucleic acids, they are wrapped around the nucleoprotein. Nucleic acid plus protein, that is nucleoprotein, and they will form a histones. Histones. So there are basically two type of nucleic acid. There are basically two type of nucleic acid. First one is nothing but the deoxyribonucleic acid. Deoxyribonucleic acid, right? So we call it as a DNA. I hope I am audible to you. Audible hai na? Now deoxyribonucleic acid or DNA, in the name itself you can clear few things. Deoxyribonucleic acid. So deoxy means deoxy sugar is there. Deoxy sugar. Deoxy means deoxy sugar. Deoxy sugar. One less oxygen means oxy sugar and deoxy sugar. What is the difference? Deoxyribose and ribose. For example, ribose is a pentose sugar mostly found in the nucleic acid. We have seen in the carbohydrates. While we was discussing about the carbohydrates, we have seen the triose, tetrose, pentose, hexose, heptose. So out of this, ribose is a pentose sugar. Ribose is a pentose sugar. Now ribose and deoxyribose. What is the main difference between these two? So deoxy means one oxygen less. So there is one less oxygen in the deoxyribose sugar and one, okay, more oxygen in the ribose sugar. Okay. Now deoxyribonucleic acid. Now, nucleic acid are nothing but they are the polymer of your nucleotides. Nucleic acids are nothing but the polymer of nucleotides. We are going to see that in upcoming part. Deoxyribonucleic acid means in this deoxyribose sugar is, and in the ribonucleic acid RNA. Ribonucleic acid means RNA. These are the two main nucleic acids present in the humans. And ribonucleic acid known as a DNA. Ribonucleic acid is known as RNA, and deoxyribonucleic acid known as a DNA. These two are the main nucleic acid. Now DNA was discovered by one Swiss scientist known as a Michner, okay? Franklin Michner, known as a Franklin Michner. So he was a Swiss scientist, Franklin Michner. He discovered the DNA, but the demonstration that DNA consists of a genetic information, हर एक का इंडिविजुअल जेनेटिक मेकअप अलग अलग है कि आपकी नाक थोड़ी बड़ी होगी, किसी की नाक थोड़ी छोटी होगी, किसी की लंबी होगी, किसी के बाल, है ना, हर एक का जेनेटिक मेकअप अलग अलग है, because everybody has different genes. Now these genes, they are nothing but the nucleic acid. So there are two type nucleic acid के दो टाइप होते हैं. पहला होता है डीऑक्सीराइबोन्यूक्लिक एसिड, इसमें डीऑक्सीराइबोस शुगर है. राइबोन्यूक्लिक एसिड इसमें राइबोस शुगर है. ठीक है. डीऑक्सीराइबोन्यूक्लिक एसिड डबल स्ट्रैंडेड मोस्टली होता है. राइबोस सिंगल आरएनए जो होता है वो मोस्टली सिंगल स्ट्रैंडेड होता है. बहुत सारा डिफरेंस है, we'll see in upcoming part. Okay, these are the two these are the two principal nucleic acids. Now DNA was discovered by Franklin Michner, and demonstration that DNA carries a genetic material was made by three scientists in 1944, known as Avery, McLeod and McCary. DNA consists of genetic information. Now this demonstration was made in 1944 by these three scientists, Avery, McLeod and McCary. Now they got Nobel Prize for this Nobel work. Obviously अगर आप कुछ नोबेल वर्क करते हो तो आपको नोबेल प्राइज भी मिलेगा, बट वो वर्क नोबेल होना चाहिए. So Avery, McLeod and McCary, they demonstrated that DNA carries a genetic information. They got a Nobel Prize in I think 1960. What is DNA and RNA? They are the chemical basis of hereditary. Okay? They are the chemical basis of hereditary or genetic information, or they are nothing but the measure of genetic information. Okay, they are nothing but the measure of genetic information. They are nothing but the chemical basis of heredity. So whatever characters that you have from your parents, from your grandparents, okay, parents, grandparents to you, and whatever character will your child will have. So that depends on your genetic makeup. That depends on your parents' genetic makeup. Okay, that depends on your grandparents' genetic makeup. So there is a something which is carried. There is something which is, okay, carried from your grandparents to parents to you, and you to your child. Now that is the genetic. That is nothing but the genes. Now DNA and RNA nucleic acids, they are the chemical basis of genetics. They are the genetic information, whatever genetic makeup is there, that resides on the DNA and RNA. Now DNA is organized in the genes. DNA is organized in genes, which is a functional unit of your genetic information. So genes are nothing but the functional unit of your genetic information. Now the relationship between DNA, RNA and protein. DNA, RNA and protein का जो relation होता है उनको अपन बोलते हैं central dogma of your life. Central dogma of life. So whatever relation between DNA, RNA and proteins, say for example DNA, then RNA, and then protein. So replication of DNA means formation of duplicate copies of your genetic material. Formation of duplicate copies of your DNA is known as a replication. DNA will be replicated. There will be a lot of copies of DNA. Okay? So replication means Replication means multiple copies of DNA, formation of multiple copies of DNA by the enzymes known as a replication. Now from DNA, RNA is synthesized. From DNA, RNA is synthesized. That process is known as a transcription. First, DNA, multiple copies of DNA will be formed, that is known as a replication. Multiple copies of DNA is formed, that is known as a replication from parent DNA. And DNA से RNA जो बनता है, so that process is known as a transcription. And from RNA, so whatever genetic material is found in your DNA, so according to that, proteins are synthesized. According to that, proteins are synthesized, and that is known as a transcription. So replication, transcription, and translation. So replication means formation of duplicate copies of DNA. Transcription means from DNA, RNA are synthesized. There is transcription, and translation means expression of that RNA, expression of that genes in the form of proteins, known as a translation. So replication means formation of duplicate copies of DNA, transcription means formation of RNA from DNA, and translation means formation of proteins from RNA. So that is known as a central dogma of life. So the relationship between DNA, RNA, and protein, they are known as central dogma of your life or molecular biology. Central dogma of molecular biology or central dogma of your this DNA, RNA. This DNA and RNA, they are the polymer, as like there are their polysaccharides, they are a polymer of monosaccharides, right? Lipids are the polymer of fatty acids, right? Proteins are the polymer of amino acids. So like that, likewise that your DNA and RNA जो है वो भी polymer है, किसका है भाई? DNA, RNA, they are the polymer of nucleotides. They are the polymer of nucleotides. So in the nucleotides, you will find a particular nitrogen containing base, nitrogen containing heterocyclic ring, nitrogen containing heterocyclic ring, pentose sugar is there, then phosphate group is there. So DNA and RNA, these are the nucleic acids which are the polymer of your nucleotides. They are the polymer of your nucleotides. Now nucleotide itself is made from nucleotide 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 कब बनता है? Nucleotide itself is made from three components. You have to remember nitrogenous base, let's say adenine, guanine, thymine, cytosine. So these are nothing but the bases, nitrogenous base. Okay, some are the purines, some are the pyrimidines. Okay. Adenine, guanine, these are known as the purine bases. Okay, purine bases, thymine, cytosine, and uracil. They are known as the pyrimidine bases. So nucleotides, they are composed of a three component. First is nothing but the nitrogenous base. In this you are going to two type of nitrogenous base, purines and pyrimidines. Purines, in that you have adenine and guanine, and in pyrimidines you have cytosine, uracil, thymine. In the pyrimidines, thymine, cytosine, uracil. Pyrimidines are the three. Pyrimidines में तीन होते हैं नाइट्रोजनस बेस, है ना, थाइमिन, साइटोसिन आणि यूरासिल. और प्यूरिन्स में दो होते हैं, एडेनिन, गुआनिन. Now these nitrogenous base, they are very common. They are the common in the uh, structure of your DNA and RNA. But some other nucleic acid or derivatives of these bases, they are also there. For example, adenosine, like that, right? C3 uracil, like that. So they are present in the DNA, RNA. They are present in the particular enzymes, you know, to recognize they are present in the particular proteins. They are particularly present in the DNA to recognize the particular enzymes, you know, कि particular enzyme उसी sequence के ऊपर वो पे cut करेगा या फिर action करेगा. That's why. But most of the time or most abundantly you will find these bases: purines like adenine, guanine; pyrimidines like thymine, cytosine, uracil. Okay. Now nucleosides, they are composed of a three component. First one is a nitrogenous base. Second component is known as a pentose sugar. Second component is known as a pentose sugar. And third component is known as a phosphate. So first component nitrogenous base. Second component pentose sugar. Third component phosphate phosphate group. Now there was a question once. There was a question once. So nucleotides and nucleosides. There are two terms. न्यूक्लियोटाइड्स एंड न्यूक्लियोसाइड्स. सो न्यूक्लियोटाइड्स, इसमें टी आता है, न्यूक्लियोटाइड टाइड, टी फॉर थ्री. तो आपको याद रखना है न्यूक्लियोटाइड में क्या रहेगा, थ्री कंपाउंड्स. न्यूक्लियोटाइड मध्ये 3 कंपाउंड आहेत आणि साइड न्यूक्लियोसाइड साइड म्हणजे की मोस्टली साइड किती असतात राइट साइड आणि लेफ्ट साइड है ना आपण दोन साइड असतात असं कन्सिडर करूया न्यूक्लियोटाइड मध्ये so you will have three components in the nucleotide and you will have two components in the nucleoside the three component कौन से so obviously first one is a nitrogenous base second one is a pentose sugar second one is a pentose sugar and third one is nothing but the phosphate group so as it is there was a question in nitrogenous base plus pentose sugar plus phosphate is known as और ऑप्शन में आता है आपको न्यूक्लियोटाइड न्यूक्लियोसाइड और ऐसा कुछ और भी न्यूक्लियोसोम्स वगैरह कुछ ऑप्शन देंगे सो टाइड मींस थ्री कंपोनेंट एंड साइड मींस टू कंपोनेंट्स सो टाइड मींस नाइट्रोजनस बेस प्लस पेंटोस शुगर प्लस फॉस्फेट ग्रुप न्यूक्लियोसाइड मींस ओनली नाइट्रोजनस बेस एंड पेंटोस शुगर सो फर्स्ट न्यूक्लियोसाइड्स दे आर फॉर्म्ड सबसे पहले न्यूक्लियोसाइड्स बनाए जाते हैं नाइट्रोजनस बेस और उसमें पेंटोस शुगर ऐड किया जाता है उसके बाद में अगर उसमें फॉस्फेट लगा दिया फॉस्फेट ग्रुप इफ देयर इज अ फॉस्फोरिलेशन एंड फॉस्फेट ग्रुप ऐड करें तो बन जाएगा न्यूक्लियोटाइड अगर एक फॉस्फेट है if there is only one phosphate in the structure of nucleotide then it is a monophosphate okay it is known as a mononucleotide if there is a one phosphate it is known as a mononucleotide if there are two phosphate it is known as a dinucleotide if there are three phosphate group known as a trinucleotide okay mono dinucleotide okay let's say for example adenine dinucleotide adenine mononucleotide adenine trinucleotide like that so nucleotide will have three component nitrogenous base pentose sugar and phosphate group nucleosides will have two component that is nitrogenous base and pentose sugar and depending upon the number of phosphate groups they are further divided into like that mononucleotide dinucleotide you know let's say adenine mononucleotide AMP adenine dinucleotide you know ADP like that are you getting or not okay because there was a question on this okay and also there was a question on this who discovered the DNA that is a Franklin Michel okay genetic information is present on the DNA it is demonstrated by Avery McLeod and McCary they carried out the experiment okay central dogma of life means replication of DNA transcription of DNA from DNA to RNA is their transcription and from RNA to protein release that is known as the central dogma of life okay now nucleotides they are the building blocks for formation of DNA and RNA they are the monomeric units nucleotides they are the building blocks for DNA and RNA so you can say nucleic acid DNA and RNA they are the polymer of nucleotides they are the polymer of nucleotides DNA RNA they are the polymer of nucleotides right or you can say nucleotides are the building blocks they are the repeating unit in the DNA and RNA now nucleotides they are classified into two types purines and pyrimidines right purines and pyrimidines nitrogenous bases they are found in the nucleotides they are nothing but the adenine guanine thymine cytosine and uracil they are the most common but some substituted bases are also there rarely to recognize a particular place for a particular enzymes depending upon the heterocyclic ring or heterocyclic base they are further divided into two types adenine and guanine okay sorry pyrimidine and purines okay so in the purine you have two types purine मध्ये दोन type असतात adenine and guanine these are the two most commonly found bases in the DNA RNA which belongs to purine category so purine का दो ही example है adenine and guanine pyrimidine there are three as I told you cytosine thymine and uracil cytosine thymine and uracil these three are known as the pyrimidines so purine and pyrimidine bases they are found in the nucleotides nucleotide it is composed of three components yeah nitrogenous base plus pentose sugar plus phosphate nucleoside means only two nitrogenous base and pentose sugar okay यहाँ पे क्वेश्चन आ चुके हैं इसको now purines adenine guanine pyrimidines thymine cytosine uracil now this uracil is found only in the RNA uracil is the pyrimidine which is found in the RNA, not in the DNA. Uracil is a pyrimidine which is found in the RNA, not in the DNA. तो RNA में क्या रहेगा फिर? Adenine, guanine, thymine, cytosine. So in the DNA you have adenine, guanine, thymine, cytosine. In the RNA you have adenine, guanine, cytosine and uracil. Instead of thymine you have uracil in case of RNA. तो देखो यहाँ पे DNA में purines कौन से होते हैं? adenine, guanine. DNA में pyrimidines कौन से होते हैं? cytosine and thymine. तो RNA में क्या होता है कि purines कौन से होते हैं? adenine, guanine. pyrimidines कौन से होते हैं? वहाँ पे RNA में pyrimidines होते हैं cytosine and uracil. Thymine इसमें नहीं होता है ना. RNA में thymine नहीं होता है ना. RNA में thymine नहीं होता है. Thymine की जगह RNA में uracil. So instead of thymine pyrimidine base, RNA has a uracil. Now you have to remember this. You might have seen this part in the 12th also. Okay. Next, the most commonly found sugar in the nucleic acid, that is nothing but the D-ribose. D-ribose. D-ribose sugar or ribose sugar. Okay. So it is found in the nucleic acid. So D-ribose. Okay. So this is sugar known as pentose sugar, which is found in the RNA, ribonucleic acid. That's why ribonucleic acid. That's why ribonucleic acid. Right. Now it's deoxyform. It's deoxyform. Deoxyribose is found in the DNA. Deoxyribose sugar is found in the DNA, and ribose sugar found in the RNA. These are the two types of sugar present in the nucleic acid. Now sugars are bound to a nitrogenous base by beta N glycosidic bond. Sugars are bound to a nitrogenous base by a beta N glycosidic bond. You have to keep in the mind this bond. Now if nitrogenous base and pentose sugar is there, it is known as nucleosides. Side में तो दो होता है, side, right side, left side, nucleoside. Now attachment of phosphate group to the nucleoside. Now it will become nucleotide. So nucleotide will have three components: nitrogenous base, pentose sugar and phosphate group. Nucleoside will have two components: nitrogenous base and then pentose sugar. Now nitrogenous base and pentose sugar, they are attached to each other by beta N glycosidic bond. You have to keep in mind. Now you can see genetic information, it is present in the nucleoproteins. Genetic information is found in the nucleoprotein. Nucleoprotein मतलब क्या? Protein present in the nucleus of cell. Nucleus के cell में जो protein होता है, nucleoprotein. Now proteins, in this protein you will find a DNA and RNA. So nucleic acid plus protein, then nucleoprotein. Now histone or protamine, so small, this type of nucleoprotein, histone and protamine. Well, nucleoprotein. So in the histone and protamine you have additional prosthetic group. Now there is one enzyme known as nuclease. Enzyme होता है nuclease, वो क्या करता है? उसको digest कर लेता है. So there is one enzyme nuclease which will degrade or digest nucleoproteins. You can also digest the nucleoprotein by using chemicals. For example, aqueous ammonia you have to heat at 115 degrees Celsius, or barium hydroxide. This is the chemical agent which can break down the nucleoprotein. Now nuclease will also do same thing. Nuclease enzyme, again depending on the, there are two types. Nucleases की भी दो type होते हैं, ribonucleases, deoxyribonucleases. Ribonucleases will break down the RNA molecule, okay, and deoxyribonuclease will hydrolyze the DNA. So they will hydrolyze the phosphodiester bond. Now in the DNA you have nucleotides, in the RNA you have nucleotides. Now all these nucleotides are the repeating units for the formation of DNA and RNA. Now they are attached by phosphodiester bond. ना उनमें क्या है कि phosphodiester bond होता है, उनमें हाइड्रोजन बॉन्डिंग होती है. Nucleus will digest the nucleoprotein and you will get nucleotides. Now nucleotides will also be degraded or digested by mononucleotidase or phosphatase. Okay. Hydrolysis. Now what this mononucleotidase is? Mononucleotidase will do, it will cause hydrolysis of ester bond between sugar and phosphoric acid. तो जो न्यूक्लियोटाइड में क्या होता है कि नाइट्रोजन बेस प्लस पेंटोस शुगर प्लस फॉस्फेट, एक फॉस्फेट ग्रुप और शुगर जो होता है ना, तो दोनों में क्या होता है कि फॉस्फोरिक एसिड प्लस शुगर जब रिएक्ट होते हैं तब वहाँ पे एक बॉन्ड बनता है, उसको बोलते हैं अपन फॉस्फोडाइस्टर बॉन्ड. ओके, फॉस्फोडाइस्टर बॉन्ड, और that is hydrolyzed by mononucleotides, कोई mononucleotides or phosphatase. So it will release nucleosides. न्यूक्लियोटाइड का अगर हाइड्रोलाइसिस हो गया तो क्या बनेगा आपका न्यूक्लियोटाइड्स. मैग्नीशियम ऑक्साइड will also do same thing. Now nucleosides and plus phosphoric acid. Phosphoric acid and phosphoric acid remove each other because phosphodiester bond is broken down by mononucleotides or phosphate. Right? Now you have nucleosides. Nucleoside means two compound. First compound में उसमें रहेगा nitrogenous base and pentose sugar. Phosphoric acid जो है बाजू को निकलती है. Now again nucleotides, they are again digested by enzymes known as the nucleosidase. Purine and pyrimidine nucleosidase. What they will cause? They will break down the nucleoside. So whatever bond between nitrogenous base and pentose sugar is there, that is broken by these nucleosides. Okay? Now you will get a sugar and you will get a nitrogenous base. Nucleotides, they are made from a nitrogenous base and sugar. So purine, pyrimidines you will get, and then ribose or deoxyribose. That's how there is evidence. Okay, that's how there is evidence that nucleotide will have three component, nucleoside will have two component. ऊपर लिखा है कि न्यूक्लियोसाइड में तीन कंपाउंड है, न्यूक्लियोसाइड में दो कंपाउंड है, न्यूक्लियोटाइड में तीन है. तो किसने बोला? प्रूफ क्या है? What is the proof? What is the evidence? Evidence is like this: if you hydrolyze the nucleoprotein, you will get a nucleotides. You will get a nucleotides. If you hydrolyze the nucleotide by mononucleotidase or phosphatase enzyme, you will get a nucleosides. Again, if you hydrolyze the nucleosides, okay, so if you hydrolyze the nucleotides, there is a release of phosphoric acid. Okay, and if you hydrolyze the nucleotides, there is a release of these nitrogenous bases and sugars. So that is the evidence. Are you getting or not? Okay. Now most commonly found nitrogenous bases in the DNA and RNA. So they are nothing but the purines. Okay, purine. Purines are nothing but the adenine and guanine. Purine के दो टाइप होते हैं, adenine, guanine. So purine का structure देखो. Now purines are numbered anti-clockwise. Purines are numbered anti-clockwise. Say for example, यहाँ पे 1, 3 like that. And pyrimidines जो है, they are numbered clockwise. है ना? 1 and 2, 1 and 3. So purines, they are numbered anti-clockwise. Pyrimidines, they are numbered clockwise. Right? Now in the purine you have adenine. So you can see adenine यहाँ पे 1, 2, 3, 4, 4 amino purine. 4 amino purine is adenine. Right? Guanine is nothing but the 2 amino 4 purine, 2 amino purine 4 1 ना, ये इसका इफेक्ट नेम बाद में भी आएगा आपको. In pyrimidine you have cytosine, you have thymine and uracil. So these are the different bases found in the nucleic acid. You can see here the structure of your sugar, pentose sugar. Structure of your pentose sugar, deoxy sugar and ribose sugar. So the difference between ribose sugar and deoxyribose sugar, that mainly is there in a carbon number 2, right? So this one is carbon number 1. It is known as a functional carbon. ये carbon number 2 होता है आपका. Right? So you can see D-ribose. D-ribose will have hydroxyl group at carbon number 2. In the deoxyribose, we will not have a hydroxyl group at carbon number 2. That is the difference between deoxyribose sugar and ribose sugar. डिफरेंस कहाँ पे होता है? कार्बन नंबर 2 जो होता है वहां पे राइबोस शुगर में हाइड्रोक्सिल ग्रुप होता है, डीऑक्सीराइबोस में नहीं होता है. डीऑक्सी नाम में रखा है ना, आप बोलते हो नाम में क्या रखा है? डीऑक्सी. डी मतलब रिमूवल, ऑक्सी मतलब ऑक्सीजन. तो उसमें ऑक्सीजन रिमूव किया है, कहाँ पे किया है भाई? कार्बन नंबर 2 के ऊपर किया है. दैट्स व्हाई डीऑक्सीराइबोस, राइबोस एंड डीऑक्सीराइबोस. व्हाट इज सेंट्रल डॉग्मा ऑफ लाइफ? सो DNA, your genetic information, your genetic makeup, that is a replicate. Okay, multiple copies of DNA, they are formed in your body, multiple copies of DNA, they are formed in the body from parent DNA. So whatever parent DNA is there, formation or synthesis of multiple copies of DNA. Simply copying of your DNA is known as a replication process. Is known as a replication process. Now after replication in your body from that DNA, okay, DNA is a mostly double stranded. Double stranded. इसमें दो strand होता है, double stranded DNA. Then from DNA there is a DNA is from DNA there is a synthesis of RNA in your body known as a transcription. Synthesis of RNA from DNA is a transcription, and finally from RNA there is a synthesis of proteins, and that process is known as translation or protein synthesis. Translation or protein synthesis. So replication, transcription, and translation. It is known as a central dogma of life or central dogma of life. Okay? Central dogma of life or central dogma of molecular biology. Central dogma of life. Replication, transcription, translation. Because we are going to see all these processes in upcoming lectures. Because replication, transcription, translation. If you don't know how there is a replication, how there is a transcription, how there is a translation, what are the different enzymes required over there. If you understand this biochemistry, these steps, definitely you are going to understand the mechanism, and also most of the antibiotics they will prevent the replication, most of the antibiotics they will prevent the transcription, they will prevent somehow some steps the replication, transcription, translation. So you have to understand how there is a replication, how there is transcription, how there is translation. Once you understand this process, you will understand the antibacterial drugs. So obviously this replication, transcription, translation, it is a different in the humans and in the microbes. So प्रोकैरियोटिक बैक्टीरिया में अलग होता है, यूकैरियोटिक बैक्टीरिया में अलग होता है, थोड़ा सा डिफरेंस होता है. Replication, transcription, translation. First, that's why we are discussing the biochemistry, and then we'll discuss the question. इसके ऊपर कैसे आता है? Now, DNA has a two strand. This is known as a one. Okay, it's one strand. This is a two strand. Second strand. DNA is having two strands. DNA is having two strands. Now these strands they are anti-parallel. They are the anti-parallel. These two strands they are anti-parallel to each other. जैसे मान लीजिए यहाँ पे है 3. This one is a 3 dash end. This 3 dash end. Okay. So here will be 5 dash end. 5 dash end. Now okay, the one strand is running from 3 dash to 5 dash. 3 dash to 5 dash. One strand will run from 5 dash to 3 dash. So wait, so these two strands they are anti-parallel, है ना? They are the anti-parallel. एक strand 3 dash to 5 dash run कर रहा है ना? और एक strand the 5 dash to 3 dash run कर रहा है ना? यहाँ पे 5 dash. So one strand is running from 3 dash end to 5 dash end. One strand is running from 5 dash to 3 dash end. So you can see these DNA strands they are exactly like a road, है ना? देखो road होता है. रोड में दो पार्ट होता है, एक लेफ्ट पार्ट एक राइट पार्ट. तो जो बड़ा वाला हाईवे होता है उसमें एक साइड से गाड़ी जा रही है और दूसरी साइड से गाड़ी आ रही है. सो डीएनए स्टैंड्स दे आर अरेंज्ड, दे आर रनिंग exactly like a your traffic road. Okay, so in the traffic road, same road carries the vehicle which is going away and some vehicle which is coming towards, okay, in opposite direction. That's how your DNA will look like the same. DNA, in the DNA, two nucleotide strands. Two nucleotide strands they are running anti-parallel. They are twisted exactly like a twisted ladder. जैसे आप रस्सी होती है, रस्सी देखो आप कैसा होता है, वो ट्विस्टिंग होती है रस्सी. So likewise that your DNA has a these two nucleotide strand, polynucleotide strand, they are running anti-parallel to each other, and they are there in a twisted manner, like a twisted rope or twisted ladder, right? Twisted ladder. Now in between that these base pairs are there. In between that these base pairs are there. So these base pairs. Okay. Now this one is known as a major group. This one is known as a major group. जो है इतना part, it is known as a major group. Major group. We'll explain it upcoming part also. उसमें distance कितना होता है सब कुछ. You can see the nucleotides, whatever nucleotides like adenine, guanine, thymine, cytosine. So these nucleotides with the pentose sugar with the and phosphate group. Okay. Now whatever nitrogenous base are there. So nitrogenous base, they say adenine, guanine, thymine, cytosine, uracil. So out of that also most of the guanines, they there is a isomerism in the nitrogenous base. What type of isomerism? There is known as tautomerism. Tautomerism. Now how most of the pyrimidines and one purine adenine. Most of the purines, not adenine, guanine. Guanine form pyrimidine and guanine form adenine. Guanine form purines. Guanine form purines and baki thymine, cytosine, uracil from your pyrimidines. All these will have two forms. Okay? At a physiological pH, जो आपका normal body का pH होता है, physiological pH. At a physiological pH, these nitrogenous bases they appear as a keto form. यहाँ पे कीटोन ग्रुप रहेगा जैसे यहाँ पे most okay, at a physiological pH they exist as a keto form. So it is known as a lactam form. Keto form is known as lactam. So most of the bases they are at a keto form in the at a physiological base, at a physiological pH. Yeah, physiological pH में वो keto form में रहते हैं. Okay, but there is a another form known as a enol form. Known as a enol. यहाँ पे देखो वो is ketoenol form of purine and pyrimidine bases. Ketoenol form of purine and pyrimidine bases. That is known as tautomers. Tautomers. Lactam and lactin. This one is not lactin. Known as enol. So ketoenol forms. So isomerism in the nucleotides. Isomerism in the nucleotides or isomerism in the nitrogenous bases. जो भी एडेनिन गुआनिन थायमिन साइटोसिन है तो उसमें का एडेनिन तो नहीं दिखा रहा है टॉटोमेरिज्म क्योंकि एडेनिन में वहाँ पे ये नहीं होता एडेनिन में वहाँ पे OH ग्रुप नहीं होता गुआनिन थायमिन साइटोसिन एंड यूरासिल one purine and three pyrimides they will show isomerism or that is known as the tautomers. They are the tautomers of each other. Now enol and ketoenol form of each other. Ketoenol form of nucleotide bases they are known as the tautomers of each other. Right. Now you can see over here how there is a formation of nucleotide nucleoside. Oh, there is a formation of nucleoside. So example is nothing but the adenosine. So this is a purine base. Purine base known as adenine. This one is a pentose sugar. This one is a pentose sugar. Ribose sugar. Okay. So ribose sugar you have first carbon, two carbon, three carbon, four carbon, and this one is the fifth carbon. Now there is fifth carbon has a CHOH group. Now whatever phosphoric acid is there. So if phosphoric acid is attached, well, if it is stratified with this iodized, well, mostly यहाँ पे ही stratification होता है. Then it will become a nucleotide. Okay. Now presently it is a nucleoside because you have nitrogenous base over here, you have pentose sugar over here. Now, nitrogenous base and pentose sugar they are attached to each other by a glycosidic bond. So which bond? Beta ion glycosidic linkage. Position of 9 of purine base at that is adenine and first carbon of your ribose sugar. तो यहाँ पे न्यूक्लियोसाइड जो है कैसे बनता है न्यूक्लियोसाइड बनता है आपका जो भी नाइट्रोजनस बेस है और जो भी आपका पेंटोज शुगर है दोनों के बीच में क्या होगा ग्लाइकोसिडिक बॉन्ड फॉर एग्जांपल लाइक तो नाइट्रोजनस बेस एडेनिन एंड योर पेंटोज शुगर राइबोस they are bound by a glycosidic bond. Okay? They are bound by glycosidic bond. One is a nucleoside. Now if there is a phosphate addition यहाँ पे अगर phosphate ग्रुप ऐड हो गया यहाँ पे कार्बन नंबर 5 क्यूब CH2OH और यहाँ पे phosphate ऐड हुआ so it will become a nucleo central dogma of your life again you can see DNA जो होता है that is a deoxyribonucleic acid this genetic information is known as a individual DNA is different हर एक का DNA अलग होता है now formation of copies of DNA parent DNA by using the one process known as a replication. So replication is the process where identical DNA is synthesized in a body or they are made by parent DNA. Okay, by using the enzymes. Synthesis of RNA or formation of RNA, or manufacturing of your RNA from the DNA, that is known as a transcription. Okay, or the transcription is nothing but the formation of or it is a process of formation of RNA from DNA. Okay, by using again the enzymes. By using again the enzymes. Then from RNA, synthesis of protein. Proteins are synthesized from RNA. From RNA, synthesis of protein is done by using translation process. Translation process क्या होता है कि protein synthesis from RNA again with the help of enzymes. Proteins में भी either ना proteins का different रोल होता है. It may be the structural protein ना functional protein, enzymes हो गए, hormones हो गए. So this is how there is a central dogma. Next is nothing but now these nucleotide bases. Nucleotide bases, for example, principal bases of nucleosides and nucleotides. पहला बेस है आपका नाइट्रोजनस बेस कौन सा है एडेनिन इज डेंटेड बाय ए राइट नाउ इसका राइबो न्यूक्लियोसाइड मतलब दो कंपाउंड है ना नाइट्रोजनस बेस प्लस पेंटोस शुगर इट इज नोन एज एडेनोसिन सो इफ एनीबॉडी आस्क यू व्हाट इज एडेनोसिन एडेनोसिन क्या है कि न्यूक्लियोसाइड है क्योंकि उसमें दो कंपाउंड है न्यूक्लियोसाइड में दो कंपाउंड है एम आई ऑडिबल टू यू ऑडिबल आएगा adenine is nucleo it is a nitrogenous base और adenosine is a nucleoside इसमें दो कंपाउंड है adenine nitrogenous base है और पेंटोस शुगर है what is a nucleotide means यहाँ पे phosphate group भी ऐड होगा ribonucleotide so adenine 5' monophosphate means एक ही phosphate group है यहाँ पे फिफ्थ कार्बन एटम जो है शुगर का राइबो शुगर का वहाँ पे फॉस्फेट ग्रुप ऐड हो चुका है फॉस्फोरिलेशन हो चुका है उसका एडेनिन 5' मोनोफॉस्फेट इज अ न्यूक्लियोटाइड मोस्टली कॉमन फॉस्फोरिलेशन ऑकर्स एट 5' मोस्टली 5' शुगर का जो फिफ्थ कार्बन एटम है उसके ऊपर ही फॉस्फेट ग्रुप ऐड होता है मोस्ट ऑफ द टाइम जहाँ पे नहीं है वहाँ पे फिर डिनोट किया जाता है 3' है ना 3' फॉस्फेट 2' mostly it is a formation of formation of phosphatized bond at only 5' position. So adenosine 5 monophosphate also known as adenylate also known as adenylate is abbreviated as AMP. AMP मतलब क्या adenosine monophosphate AMP adenosine monophosphate और adenylate then again guanine is a nitrogenous base उसका न्यूक्लियोसाइड क्या होगा गोनोसिन और उसका न्यूक्लियोटाइड क्या होगा गोनोसिन 5' मोनोफॉस्फेट और गोनाइलेट गोनोसिन 5' मोनोफॉस्फेट और गोनाइलेट उसको अपन एब्रिविएट करेंगे जीएमपी जीएमपी मतलब गुड मैन्युफैक्चरिंग प्रैक्टिस नहीं है साइटोसिन अनदर बेस होता है नाइट्रोजन बेस साइटोसिन सो इसका न्यूक्लियोसाइड क्या होगा साइटिडीन न्यूक्लियोसाइड विल बी साइटिडीन cytosine plus your pentose sugar cytidine and if you add a phosphate so it will get a cytidine 5 monophosphate or cytidylate then next होता है आपका नाइट्रोजन बेस यूरासिल so it will form uridine nucleoside and nucleotide will be uridine 5' monophosphate that is a uridylate abbreviated as a UMP then you can see base ribonucleoside deoxyribonucleoside 5 monophosphate abbreviation then first adenine so deoxyadenine ये इसमें राइबो शुगर था इसलिए है ना पहले इसमें क्या था राइबो शुगर ये आरएनए में रहते हैं so in the RNA you have adenine guanine cytosine and uracil these four bases so these ribonucleotides they are found in the RNA इसमें सब इसमें रहेगा ये सब रहेगा RNA में now DNA adenine guanine cytosine and thymine so adenine will form a deoxyadenosine ribonucleoside and it will form a deoxyadenine 5' monophosphate that is known as deoxyadenylate or DAP. So this one is a nucleotide. Guanine will form a deoxyguanosine nucleoside in the DNA. Deoxyguanosine 5' monophosphate or deoxyguanylate that is a nucleotide. Now cytosine will form a deoxycytidine as a nucleoside and it will form a deoxycytidine 5' monophosphate or deoxycytidylate that is a DCMP as a nucleotide. Thymine will form a deoxythymidine as a nucleoside, it will form a deoxythymidine 5' monophosphate or deoxythymidylate as a nucleotide, that is DTMP. They are attached like this. You can see over here. तो ये जो structure है adenine का है, is adenine purine. So this is the ninth carbon, so it will form a glycosidic bond with the first carbon atom of sugar. Sugar first, second, third. तो CH2OH, जो OH होता है वो आपको phosphate को प्रोड्यूस करता है. It has three components: resonance base, pentose sugar, and phosphate nucleotide. Adenine mononucleotide phosphate. Adenine mononucleotide AMP. Adenine monophosphate. Now this is thymine monophosphate. Thymine monophosphate. So formation of nucleotides over here. Right? Now this wait. Now these bases or these nucleotides are nothing but they are the building blocks for your DNA and RNA structure. का काम होता है कि ये DNA RNA में रिपीटिंग यूनिट्स होते हैं. रिपीटिंग यूनिट्स ऑफ न्यूक्लियोसाइड्स आर प्रेजेंट इन द DNA RNA. ये तो एक काम हो गया. They are the components of DNA, but they have another role also. So they have another role also. Now, regulation of cell activities extra, like EAP, cyclic GMP. They also regulate the cell activities. They also are having different role in the synthesis. For example, synthesis of uric acid. So xanthine, hypoxanthine, uric acid. They are also purines. So nucleotides, they are not only having a part of DNA, but also in the body they have different role. Nucleoside में सिर्फ DNA में नहीं होता है. They are not only repeating polymer of DNA, but also they have a different role in the body. कोई याद रखना है. तो adenine का name होता है 6-aminopurine. Adenine is nothing but 6-aminopurine. 6-aminopurine. 6-aminopurine कैसे? So 6-aminopurine. That is adenine. Adenine is 6-aminopurine. Number है बाद में देख लो number. Then guanine is a 2-amino-6-oxypurine. Oxy or oxo इसमें कीटोन है. 6-oxypurine. 2-amino-6-oxypurine. तो guanine और adenine में क्या फर्क है? Only difference is that in a guanine there is a keto. That's why adenine जो है ना, it will not show tautomerism because there is no keto group in this. But guanine will have ketone group, so this ketone group may be converted into OH group, ketoenol. Guanine can form a ketoenol form, so it can show tautomerism, but adenine will not show. In the cytosine also you can see it is a pyrimidine base. In a pyrimidine 1, 2, 3, 1, 2, 3, 4, 5, 6. 2-oxo 4-aminopyrimidine. 2-oxo 4-aminopyrimidine. That is cytosine. And this cytosine you will have one ketone group, so it will form a keto and enol form. So that's why tautomerism. Next one is the thymine. Thymine again the 1,2,4-dioxo. Okay, 2,4-dioxo 5-methyl pyrimidine. That is thymine. Uracil क्या है? Is again 2,4-dioxo-pyrimidine. 2,4-dioxy-pyrimidine. So all these except adenine, all the other purine and pyrimidine bases. Purine तो एक ही उसने होनी. बाकी pyrimidine base, they will show the ketoenol tautomerism. Ketoenol tautomerism. Except adenine, all other bases found in the DNA RNA, they will show ketoenol tautomerism. You have to remember. Now structure of DNA and RNA. Structure of DNA and RNA. So you know that DNA is nothing but the polymer. DNA RNA are nothing but the polymer of nucleotides. So particularly DNA is a polymer of deoxyribonucleotide. Here chains are there. 2 strands are there. Chain को ही अपन strand बोलते हैं. There are 2 chains. They are held together by 3'5' phosphodiester bridge or phosphodiester bonds. So we say 3'5' phosphodiester bridge. 2 strands of DNA, they are held together by 3'5' phosphodiester bridges. So by hydrogen bonding. The backbone is nothing but the hydrogen bond. The backbone is nothing but the hydrogen bond. Right? So DNA is a polymer of deoxyribonucleotide held together by 5'3' the phosphodiester bond. In the DNA, there is one rule known as the Chargaff's rule. Chargaff's rule. Chargaff's rule of DNA composition. So according to this rule, so Irvin Chargaff in 1940, scientist का नाम है Irvin Chargaff 1940. He proposed that there is a equal number of adenine and thymine residue in the DNA and equal number of cytosine and guanine residue. तो ये चारकॉप महाशय जो थे उन्होंने क्या देखा ना, ये अपने को ताप रहता है, मतलब साइंटिस्ट है ये, कुछ ना कुछ देख के आते हैं, कुछ ना कुछ समझते हैं, फिर अपने को तकलीफ एडेनिन एंड थायमिन का कहना है कि डीएनए में एडेनिन और थायमिन जो है ना वो इक्वल होता है, जितने एडेनिन है उतने थायमिन है, और उसके जैसे ही जितने ग्वानिन है डीएनए में उतने क्या है साइटोसिन है. सो उनका मतलब है कि A is equal to T, AT and GC. This ratio is fixed in the DNA. So in the DNA, number of adenine is equal to the number of thymine. Number of guanine is equal to number of cytosine. So this rule is known as the Chargaff's rule. Now DNA will follow this rule. DNA will follow this rule. That's why DNA is a double stranded structure, and DNA will follow this rule. But see, RNA is a single stranded structure, and RNA will not follow Chargaff's rule. But double stranded RNA जो होता है, definitely that will follow a Chargaff's rule. Normal RNA will not follow Chargaff's rule, but double stranded RNA is a single stranded, but sometimes there are double stranded RNA also. Okay? So that RNA will follow the rule, but normal RNA will not follow. Single stranded RNA will not follow the Chargaff's rule. DNA will follow the Chargaff's rule. Okay? It is known as a Chargaff's rule of molar equivalence between purines and pyrimidines. और भी क्वेश्चन आ चुके already. Chargaff's rule is related to. Chargaff's rule is nothing but the molar equivalence between purines and pyrimidines. As per the rules, it states that there is a equivalence as far as the molar is concerned. Okay, there is a equivalence between the number of moles of purines and thymine. Okay, this means the equivalence in the moles of adenine and thymine in the DNA, that like exactly like this guanine and cytosine. तो जितने एडेनिन होंगे उतने थायमिन होंगे डीएनए में, जितने गुआनिन होंगे जितने ही साइटोसिन होंगे. ये इरविन चारगाफ महाशय जो थे उन्होंने बोला था, तो उसके बेसिस पे उनको बोलते हैं अपन चारगाफ्स रूल. So it is a rule of molar equivalence between purines and pyrimidines in DNA structure. क्वेश्चन आता है इसके ऊपर, एक इंपॉर्टेंट दे आ चुका है. Now DNA is a double stranded, right? Double helical structure of DNA. Double helical structure of DNA. It was proposed by James Watson and Francis Crick. James Watson and Francis Crick in 1953. He will get a Nobel Prize in 1960 or 62. It is known as the Watson and Crick model. Double stranded DNA जो होता है वो Watson and Crick model से नाम के जाना. Now, as per them, DNA is made from two polydeoxyribonucleotide chain. Two polydeoxyribonucleotide chain. Genetic information. ये chain में क्या है? दो chain. So there are two strands or two chains of polydeoxynucleotide. Polydeoxynucleotide के दो chain है, दो strand है. और ये जो दो chain है या फिर दो strand है, तो एक strand को बोलेंगे अपन एक strand को बोलेंगे अपन template strand और एक को बोलेंगे अपन complementary strand. So there are two strands in the or two chain in the nucleic DNA. So one chain or strand is known as a template strand or chain. Second one is known as a complementary strand or chain. Now genetic information is present in only one strand. Okay, that is known as a template strand. Genetic information is present in the template strand. ये आपको याद रखना. The DNA you have two strands, that is a template strand and complementary strand. So genetic information is found on the template strand. Genetic information resides on the template strand, and these two strands, template strand and complementary strand, they are anti-parallel to each other. Means they are running in opposite direction. One will be 3 dash to 5 dash. Second will be 5 dash to 3 dash. They are anti-parallel to each other. ये क्या रखना है? Now these two strands, they are held together by hydrogen bonds. ये मैंने पहले भी बोला था. These two strands, they are held together by hydrogen bonds. Now each turn of double helix distance is nothing but around 34 and so on. So each turn or each helix or each groove of DNA, double helical DNA, is of 34 and so on. In each helix or in each groove, there are 10 nucleotide pairs. There are 10 nucleotide pairs. Each pair is placed at a distance at 3.4 angstrom. तो जो मेजर ग्रुप होता है मेजर वाला ये जो होता है डीएनए का उसका डिस्टेंस कितना होता है 34 एंगस्ट्रॉम उसका साइज एंड उसमें 10 बेस पेयर आते हैं तो विद द 10 बेस पेयर ऑफ 10 पेयर्स ऑफ न्यूक्लियोटाइड्स एक ग्रुप में 10 पेयर रहेंगे न्यूक्लियोटाइड के ईच पेयर इज प्लेस्ड एट अ डिस्टेंस ऑफ 3.4 एंगस्ट्रॉम and the width of double helix is nothing but 20 angstrom. कभी भी इसके ऊपर क्वेश्चन आता है. Are you getting or not? समझ रहे आपको? Are you getting or not? समझ रहे हैं? Very easy. We'll see one more slide and then we'll take tomorrow. Double helical structure of DNA. There are two three types of double helical structure of DNA. DNA का जो helical structure है उसका भी type होता है. तो इसके ऊपर क्वेश्चन आ चुका है. Which is the most common helix, most common double helix form of DNA? BDNA, known as a bent DNA, or also known as a right-handed DNA. DNA जो होता है उसको ही bent DNA बोलते हैं, उसको ही right-handed DNA बोलते हैं, and that is the most common of DNA found in the genetics, and that is the model which was proposed by Watson and Crick. Watson and Crick का जो DNA का मॉडल है कौन सा है BDNA. मोस्ट कॉमन भी BDNA होता है. BDNA is the right-handed DNA. That is model proposed by Watson and Crick. It is the most common. Now another form of DNA, ADNA is known as a right-handed DNA again. Then another type is there ZDNA. ZDNA is known as a left-handed DNA. There are three types of DNA. एक होता है BDNA, that is the most common. दूसरा होता है ADNA, तीसरा होता है ZDNA. BDNA को भी bent DNA बोलते हैं अपन, राइट हैंडेड DNA लिख सकता है और वैटसन एंड क्रिक का मॉडल है. में ADNA होता है वो भी राइट हैंडेड होता है but that is not common. Okay, then ZDNA is there which is a left handed DNA. एक बार क्वेश्चन ऐसा आया था लेफ्ट हैंडेड DNA, identify the left-handed DNA from the following. तो ZDNA क्या होता है लेफ्ट हैंडेड DNA. Okay, ADNA and BDNA दोनों भी क्या है राइट हैंडेड DNA. And out of that most common कौन सा है BDNA. BDNA ही को अपन बोलते हैं वैटसन एंड क्रिक मॉडल. तो दो लाइन है यहाँ पे सिंपली बट लेकिन ये दो लाइन के ऊपर ही दो बार क्वेश्चन. ZDNA में क्या होता है कि the polynucleotide chains they moves in a zigzag fashion, so that's why ZDNA. ZDNA में क्या है the chain of polynucleotide that moves in a zigzag fashion, that's why ZDNA. BDNA is most prominent under physiological condition. जो होता है most prominent और most common होता है under physiological condition. Other forms of DNA like है ना, ये तो डबल स्टैंडर्ड DNA हो गया आपका, डबल स्टैंडर्ड DNA. But other forms are there like bent DNA, triple stranded DNA and four stranded DNA is also. याद रखना आपको BDNA is a Watson and Crick model. It is a right-handed. ADNA is also right-handed, and then your ZDNA is a left-handed. BDNA is the most prominent under physiological condition. Other forms कौन से bent DNA, triple stranded DNA is there, four stranded DNA is also there. Now denaturation of DNA, loss of helical structure of DNA is known as a denaturation of DNA. The helical structure can be denatured, okay, or it can be denatured by, it can be measured by UV spectroscopy at 260 nanometer. तो ये denature कर सकते हैं. You can denature the DNA at 260 nanometer. अगर ये वेवलेंथ का UV में रख दिया सॉल्यूशन अपने DNA का तो वो denatured होगा. Denaturation means what? Loss of helical structure of DNA. जो हेलिकल स्ट्रक्चर है डबल हेलिक्स वो लॉस होना, वो टूट जाना, उसमें क्या हाइड्रोजन बॉन्ड्स ब्रेक होना, उसको क्या बोलेंगे loss of helical structure of DNA is known as the denaturation. इसके ऊपर एक क्वेश्चन आ चुका है. क्वेश्चन कैसा है था? Melting temperature Tm. Melting temperature Tm is nothing but the temperature at which half of the helical structure of DNA is lost. और इसके ऊपर क्वेश्चन आ चुका है. Melting temperature of DNA. Melting temperature of DNA Tm. It is the temperature at which 50% of DNA structure is lost. Known as the melting temperature of DNA. Melting temperature of DNA or Tm is nothing but the temperature at which half of the helical structure of DNA is lost, known as the melting temperature. Okay. Phenomenon of increase in the absorption of purine and pyrimidines following the denaturation is known as the hypochromicity. Hyperchromicity. Denaturation होता है DNA का. So in the DNA double base, purines, pyrimidines, they are attached, they are bound by hydrogen bonds. Now due to denaturation, there is a breakdown of hydrogen bonds and release of individual nucleotides. 3. तो nucleotides individual release हो गए तो यूवी में absorption है ना, intensity जो है तो absorbance जो है वो बढ़ाएगा. That's why known as hyperchromicity. So structure of DNA. Do you understand? समझ रहे हैं आपको DNA यार? DNA में क्या क्या है क्या नहीं है? और the structure of DNA. Structure of DNA again. DNA is a double helix. There are two strands, है ना? So each strand they are they are running anti-parallel. Say एक strand, per of one strand. So here per. So lastly आएगा इसका three here. What is this? If this is a second strand known as a 3', so here per. These two strands they are anti-parallel to each other. Now these, so this one is known as a one groove. This one is known as a one groove. One groove. So either it may be my major groove. ये तो major हो गया अभी. और this one is a minor groove. ये वाला groove जो है वो minor है. ये वाला जो है major है. और now the distance between these two grooves, okay, so known as is a 4 angstrom or 3.4 nanometer, and the distance between two nucleotide, ये दो nucleotide है उसके बीच में ये distance है, it is 0.34 nanometer or 3.4 angstrom. These are the anti-parallel strand. Okay, the size of the width of this DNA is nothing but the 20 angstrom. 3.4 nanometer मतलब 3.4 angstrom. 34 angstrom. Phosphate bond form a ray. Phosphate bond form a ray means there is a phosphatized bond between base pairs. Okay, between base pairs. The backbone is nothing but the hydrogen bonds. It's known as a double helical structure of DNA or Watson and Crick model, known as a PDA. Formation will like this. Say adenine and thymine, ATGC, adenine and thymine. There is a hydrogen bond over there. Cytosine and guanine, they will form a pair. Adenine, guanine, they will form a pair. Cytosine and adenine, thymine will form a pair in the DNA, and cytosine and guanine they will form a pair in the DNA. So adenine क्या होता है? Purine. क्या है? Pyrimidine. Pyrimidine base. So A is equal to T है ना, G is equal to C. मतलब जितने एडेनिन होंगे डीएनए में उतने ही थाइमिन होंगे और जितने साइटोसिन होंगे उतने ही गुआनिन होंगे. इस बेस पेयर विल फॉर्म हाइड्रोजन बॉन्ड्स. Now structure of RNA. RNA is nothing but the polynucleotide of, again RNA is nothing but the polymer of ribonucleotide. से DNA क्या था? It is a polymer of deoxyribonucleotide. तो RNA क्या रहेगा? RNA is nothing but the polymer of ribonucleotide held together by same thing, phosphodiester bond or bridges. Now there is a difference in the DNA and RNA. So there is a main difference is nothing but the sugar. तो जो DNA होगा उसमें deoxyribose sugar होगा, RNA में ribose sugar. So in the DNA you have deoxyribose sugar and in the RNA you have ribose sugar. Another difference is there that basis of pyrimidine. Now in case of DNA you have adenine, guanine, thymine and cytosine. Okay? adenine, guanine, they are nothing but the purines, thymine, cytosine are nothing but the pyrimidates. But in case of RNA, you have one pyrimidine different. In case of RNA, purines are nothing but again same adenine and guanine. In case of pyrimidines, so there is no thymine. Instead of thymine, there is uracil. So it has a difference in sugar. Instead of thymine, RNA has a uracil. Okay, and usually RNA are nothing but the single strand. Chargaff's rule is not followed, not obeyed. Now RNA RNA are more susceptible for alkaline hydrolysis, and they also give color reaction with the orsinol. इतना ही important है actually. Now there are again types of RNA. Types of RNA कितने होते हैं? मैसेंजर RNA, also known as mRNA. ना टाइप्स ऑफ आरएनए, मैसेंजर आरएनए होता है, mRNA, उसका फंक्शन क्या है वो अपन देखेंगे बाद में. ओके, सो आउट ऑफ टोटल आरएनए आरएनए पूल, टोटल आरएनए जो बॉडी में है उसमें का 5 to 10% क्या होता है? सो आउट ऑफ टोटल आरएनए 5 to 10% इज mRNA. आउट ऑफ दैट देन सेकंड टाइप होता है ट्रांसफर आरएनए और टीआरएनए. So it is a 10 to 20% of total RNA count. Then third होता है ribosomal RNA, so known as rRNA, that is 50 to 80%. Why ribosomal RNA is more? Because ribosomal RNA is involved in the protein synthesis, right? mRNA will code, mRNA will carry a genetic code. mRNA में क्या होता है? genetic code होता है, genetic code. ट्रांसफर आरएनए का काम क्या है? ट्रांसफर आरएनए का काम होता है टू ट्रांसफर अमीनो एसिड फॉर द प्रोटीन सिंथेसिस. ओके, सो रिमेनिंग पार्ट विल सी टुमारो. इज दैट फाइन? ओके, आपने थोड़ा सा ना 2 hours पेशेंस, इफ यू कैन अटेम्प्ट